Okay. Do you hear me okay? Yep. Okay, good morning everybody. Today is the third day. It's the fourth. We made a long way. We have another long way. And today we will continue the story of do myself well. Today we continue the story about the environment and nanotechnology. We will start with a very interesting sub subject, which is called nanobiotechnology, which is essentially the interface between the nanotechnology and the biotechnology world, and we will see how this is affecting our understanding of biotechnology. What we will talk about today, we will discuss antimicrobial nanoparticles, how they help, our dealing with the environment. Then we will talk about carbon dots again. As you see, I really like carbon dots. This is part of my expertise. We will talk about carbon dots for bacterial imaging and degradation. And then we will describe an interesting application of carbon dots for antiviral face masks. You know, I don't think this is working well. Maybe I'll stand like this. Or I can talk with this. attached to the surfaces. Usually bacteria have a surface which is positively charged. So what you need to take metal nanoparticles and coat them with some molecules with negative charge. Then you're going to have attraction, positive bacteria, negative nanoparticles, and they bind together. And why do you want metal nanoparticles? When you have metal nanoparticles, you can apply a very useful analytical technique, spectroscopic technique called SIRS. SIRS is a technique, is an abbreviation for Surface Enhanced Raman Spectroscopy. Raman spectroscopy, for those of you who don't know, it's a very sensitive technique that gives you signal from metal regions, from metal areas. So if we have metal nanoparticles bind to bacterial surfaces and we apply SIRS, we apply Raman spectroscopy, then we have a Raman spectrum for the nanoparticles. Now each nanoparticle will have a different Raman spectrum depending also on the environment. So if the nanoparticles sit on the surface, spectrum, if the nanoparticle sits on this bacteria, we're going to have a different spectrum. That's how, by using nanoparticles, metallic nanoparticles, we can get specific Raman spectra for specific bacteria. And that's how we're going to be able to identify the bacteria and tell ourselves, here there is bacterial contamination, and here we don't have bacterial contamination. This is another example of using nanoparticles for bacterial imaging and also killing. What we do here we attach to the bacteria's surface gold nanoparticles. On the left, we attach spherical gold nanoparticles. On the right, we attach rod-like gold nanoparticles. Now, why it is important? Because in certain shapes of gold nanoparticles, and actually other types of nanoparticles, what we can do, we can irradiate with infrared light. And when you have rod-shaped gold nanoparticles, they absorb the infrared light. What is infrared light? You know? Where does it come from? From sun, but also from the body. Infrared light, in general, comes from heat. When something emits infrared, it means that it is hot. So if these nanoparticles will absorb infrared light, what it does, it simply heats the nanoparticles. Now, if you absorb infrared and you heat the nanoparticles, and you heat more nanoparticles and more, and the nanoparticle sits on the surface of the bacteria, what happens eventually? The bacteria will die. You simply burn the bacteria using IR, infrared light, shining on the bacteria, absorbing by the rod-shaped nanoparticle, and killing the bacteria. So these are very nice examples of using nanoparticles particularly metal nanoparticles, gold nanoparticles, both for imaging or for seeing bacteria through Raman spectroscopy, also killing bacteria through the thermal effect, through heating and burning the bacteria. Actually, the concept of using nanoparticle for killing bacteria, the antimicrobial metal nanoparticles, is a very big concept. It's a very big field of research. And actually, many people, and there are actually also commercial products for using silver nanoparticles as antimicrobial agent. You will actually find coatings for surfaces made from silver nanoparticles that are used for antibacterial effects. In hospitals, for example, in instruments surgeons use, often they coat them with silver nanoparticles, and this makes them very antimicrobial. The reason actually is not entirely clear, but what we think is happening is that silver nanoparticles have many electrons that are free. Why they are free? Because silver is a metal. And in metals, we have a lot of electrons on the surface of the metal. Now, if we have many electrons on the surface and bacteria coming, the electrons can oxidize the bacteria or reduce the bacteria or go into the bacteria and disrupt biochemical processes. What people found is that in silver nanoparticles in particular, this redox effect, this electronic effect is very strong. Maybe because in silver nanoparticles, the electrons are free, are more free than other nanoparticles. Anyway, the thing is that silver nanoparticles are very useful as antimicrobial agents, and you can actually design various products using silver nanoparticle for antimicrobial activity. The other thing is actually it's very easy to make silver nanoparticles, and they're very cheap, relatively speaking. That's why people use them for antimicrobial applications. These are some examples of what silver nanoparticles can do in bacteria. This is a general shape of bacteria. We have DNA, we have proteins and enzymes inside the bacteria. And here we can see what the silver nanoparticle can do. These are the silver nanoparticles. You see that they are very small. If they are very small, they can go into the bacterial cell very easily. And then when they are inside the bacterial cell, using this redox effect, the reduction oxidation effect of the electrons, they can do damage. They can damage the DNA. They can affect biochemical processes of enzymes. They can break the membrane. They can destroy other proteins, membrane proteins, or proteins inside the bacteria. Overall, they are very effective in killing bacteria. So silver nanoparticles as antimicrobial agents. Now, we talked about silver nanoparticles, but these mechanisms of antimicrobial activity are very general for many types of
nanoparticles, not only silver and actually not only metal nanoparticles. So whenever we have nanoparticles and they can go into the bacteria, inside the bacterial cells, they can do a lot of damage. They can do a lot of damage everywhere, in all the different molecules, organelles, different parts of the bacterial cell. One particularly interesting effect of metal nanoparticles is on the bacterial biofilm. Have you heard of the bacterial biofilm? You know, the reason bacteria in our body are so bad, it's not only because bacteria themselves are bad. It's because bacteria, when they go into our body, they form what's called a biofilm. A biofilm is like a shield. It's a matrix. It's like a building that bacteria build above themselves. And by doing this, it protects them from the body. You know, the body is very good in preventing infection, in preventing attack by bacteria or by viruses. How the body does that? The body has the immune mechanism. When something goes into our body, some foreign thing, viruses, bacteria, proteins, anything foreign that comes to our body, the body immediately gets into action. It secretes proteins. It has cells, white blood cells, other types of immune mechanism and immune proteins that go very fast to the foreign object and destroy it. That's why our body is very good on surviving in this world with all the bad things that are around us. So what would the bacteria do to prevent this attack by the body? What they do, they have this shield that they build around themselves. Now this shield is made of very strong proteins and sugars. So when you have this shield, this biofilm, the bacteria actually is sitting here inside this shield, and nothing goes in, and the bacteria are happy. They can proliferate. They can grow. The body cannot affect them. And after they grow, and you have millions of bacteria inside this biofilm, then you go out, and for the body, it's much more difficult to fight millions of bacteria. And then you're sick. You know where else we have biofilm? In our teeth. You know why we have holes in our teeth? We brush our teeth every morning and every afternoon maybe. But still, if you're a dentist, you have lots of work. You have holes in your teeth. The reason is biofilm. When bacteria go on your teeth, immediately they form this shield. And then when you have their shield, this biofilm, you can brush your teeth 3 times a day and it will not help. Because the biofilm protects the bacteria. So then you have biofilm in your teeth. And the bacteria are very happy. They proliferate and they start making holes in our teeth. That's exactly why we have holes. Some bacteria have big biofilm and some bacteria have small biofilm. But this is the reason or one of the main reasons why bacteria are very bad for us. But luckily, we have nanoparticles. So these are the bacteria in our teeth, the biofilm in our teeth. Now, how can nanoparticles help us with the bacterial biofilm? Now, the interesting thing is, this is another example of biofilm. The interesting thing is that some nanoparticles can penetrate this shield, this biofilm, and kill the bacteria even inside the biofilm. For example, the silver nanoparticle. Silver nanoparticles, surprisingly, they can go inside the biofilm. This is the matrix of the biofilm. They can slowly break the biofilm and reach the bacteria under the biofilm. People still don't know exactly how it happens, but when the silver nanoparticle destroy the biofilm, the bacteria are no longer protected. And that's how we can kill the bacteria, by the silver nanoparticles or the immune mechanism of the body. Now this looks very nice, but it's not so simple, because silver itself is toxic. We don't want to start swallowing silver into our body, because silver by itself might cause damage to our body. So this is, it's always positive things and negative things. If we can find a drug, a treatment that will use this silver nanoparticle effect of destroying the biofilm and still not be toxic, not be bad to our body, this will be a very interesting and effective way to fight bacteria. Let's describe another interesting application of nanomaterials, nanotechnology, for fighting bacteria. In the example here, something that we did in my lab several years ago, we used carbon dots, both for imaging bacteria and also for killing bacteria. Now, the bacteria that we studied is called Pseudomonas aeruginosa. Pseudomonas aeruginosa is a very bad bacteria. It gives us pneumonia. It's involved in many really bad infections. If you type in Google Pseudomonas aeruginosa, you will find not so nice pictures of people sick with various diseases. So we definitely want to know if people have Pseudomonas aeruginosa in their bodies, and we want to know if we can kill these bacteria, these Pseudomonas aeruginosa. These are just some of the ugly pictures that you find in the internet about Pseudomonas aeruginosa. Okay, you see somebody here with infection in the lung, just like pneumonia. Here you have some infection of the skin with Pseudomonas aeruginosa. Anyway, I wish you all good health, so you will not have Pseudomonas, but we will try to help you and people in general with identifying and killing Pseudomonas aeruginosa. Now, another very bad problem with Pseudomonas and other bacteria is something that is called multi-drug resistance. You heard this term, multi-drug resistance? You know, some bacteria develop resistance against antibiotic compounds because of evolution. We give ourselves penicillin and various other antibiotics, but the bacteria learn how to deal with that. And this is actually very dangerous because as time goes by, there is bacteria that can resist antibiotics. And then we cannot do anything to treat disease. So this is another good reason why we want to find new ways to kill bacteria, new antimicrobial agents. So here we go back to the carbon dots. Remember our carbon dots, which we talked about several times the last couple of days? Just reminding you why we like carbon dots. We like carbon dots because of the colors. And we have colors because of the surface of the carbon dots. Now, the interesting things about carbon dots is that it's very easy to control the surface of the carbon dots. We can actually put on the surface or generate on the surface various different chemical groups. And by doing that, we can have the carbon dots go into specific locations and into specific cells. How we do that? For that, we need to look more carefully on the surface of bacteria. Now, bacteria have very interesting surface properties. Bacteria which belong to the gram-negative family. There are two main types of bacteria in the world. Gram-negative and gram-positive. In both cases, you can find good bacteria and bad bacteria. Now remember when we talked about Pseudomonas aeruginosa, Pseudomonas aeruginosa belong to the gram-negative family of bacteria. Now, what we know about gram-negative bacteria is that the surface of the gram-negative bacteria have lots of sugars and also a special types of lipid which are called lipopolysaccharides. These are lipids with sugar on top. And these lipopolysaccharides are very specific to gram-negative, and even more than that, Pseudomonas aeruginosa have specific lipopolysaccharides or LPS on their surface. Now, in this project, this was very good for us because it gave us a target for binding carbon dots through the lipopolysaccharides on the surface of the bacteria. So this is the structure of LPS, lipopolysaccharide. Okay, we see the lipids here, and then there's a connector and a very long chain of polysaccharides, sugars. So what we're going to do with this? What we can do is we can make carbon dots with special surface. We will take, remember, the way we make carbon dots is that we take some carbon source and we heat it to a about 100 degrees or 150 degrees. So what we made carbon dots from two carbon sources. One of them was citric acid, what we find in lemon, okay? Very cheap, very easy. And then we took another small molecule called aminoguanidine. Now we did not need to take specifically aminoguanidine, but what we need and what aminoguanidine gave us is these units, the amino units. And when we make the carbon dots, you see that these carbon dots have the aminoguanidine units on their surface. Now the special thing about aminoguanidine is that it recognizes and it binds very strongly this part of the lipopolysaccharide. So what we accomplished is carbon dots that will bind specifically to the surface of the Pseudomonas aeruginosa bacteria through the interaction between aminoguanidine and the lipopolysaccharide. So let's see what happens. In this case, we use E. coli bacteria. Okay? We use E. coli bacteria, which is also a negative type of bacteria, gram-negative bacteria. We added the carbon dots with the aminoguanidine, and this is what we get. Remember the color of the carbon dots, the beautiful color of the carbon dots. Now what we did, we simply mixed the solution of the carbon dots with the bacteria. The bacteria actually didn't care. They just grew. But the carbon dots were attached to the surface of the bacteria through the interactions between the aminoguanidine and the LPS, and we get bacteria with color. This is not a visible color, this is a fluorescent color. And still, you can look at the microscope using a fluorescent microscope, which is also something very simple and cheap, and we can see the bacteria. So think about it. If you're sick, you go to the doctor, and the doctor can take some swab from your throat or some blood, and the doctor puts it on a plate with the carbon dots. You wait 1 hour, 2 hours, and then you look at the microscope. If you have infection, you can see the bacteria very clearly, very nice color. It means you're sick. If the doctor doesn't see anything, everything is good. And actually, what you can do not just seeing the bacteria, you can also kill the bacteria. If you put a lot of carbon dots with the aminoguanidine, the aminoguanidine carbon dots will bind to the cell, to the Pseudomonas aeruginosa cell, and kill the bacteria. Because the bacteria doesn't like so many small carbon nanoparticles bind to the surface and affect the biochemical processes on the surface of the bacteria. So these are electron microscopy images. Okay? These are the Pseudomonas bacteria. You see the rod shape. This is control without carbon dots. Here we added the carbon dots and we waited 30 minutes. You see that all these are dead cells. Carbon dots kill the bacteria. Now, this technique is very specific to gram-negative bacteria and more specifically to Pseudomonas aeruginosa. Because if you do this with another type of bacteria, this is a bacteria called Staphylococcus aureus. This is also a not so friendly bacteria. You don't want to have Staphylococcus in your body. But this is different disease. And the thing with Staphylococcus aureus, it's not a gram-negative bacteria. It's a gram-positive bacteria. And gram-positive bacteria don't have the LPS in their surface. So, carbon dots with the guanidine head group cannot bind to Staphylococcus surface. So when we mix Staphylococcus bacteria with the carbon dots, nothing happens. Because the carbon dots cannot bind to the surface of this bacteria. So what we see here is a very nice way to use carbon dots as specific drugs, specific antimicrobial agents against Pseudomonas aeruginosa. And we can use the same concept for targeting other types of bacteria because it's very easy to control the surface of the carbon dots. We can put in the surface of the carbon dots different molecules that will recognize other types of bacteria. So this is a story about antimicrobial carbon dots. Actually, we can, because it's so simple to make carbon dots, we can use them in different products to give them the antimicrobial properties. This is just a dream. Okay? There's no products yet, but it's certainly possible. We can make antimicrobial cream. Let's say you have a wound with bacterial infection, so you can make carbon dots. The same carbon dot that I showed you before, you mix it with something and you're going to have a cream. A cream which will be antibacterial. This is a story about antimicrobial carbon dots. Let me tell you another story about antiviral effect of carbon dots. Actually, this project started in the COVID days. Remember the bad days of COVID? The terrible days of COVID? Just like a nightmare, we all try to forget it. But for me at least, part of the nightmare of COVID was, I don't know how it was in India, but in Israel, for maybe 1 year, they wanted you to wear face mask. And I hate face masks. So anyway, because of this requirement to wear a face mask, we thought of this project of making special face masks with carbon dots that will help us fighting COVID and other types of viruses. And the story here actually is related to, remember yesterday when we talked about the photothermal effect of carbon dots? You shine light, you get heat. That's the simple concept that we used in this system, in the story of antiviral face masks. So this is the face mask we fabricated. Okay? We took carbon dots. Okay? We mix them with a certain polymer that will help them stick together and form a film. And then we add them to a very thin polymer layer. So we have a polymer with carbon dots inside. Now the polymer layer was very thin and used a special polymer that has pores. You see my picture? Yes. This is the polymer. This is this polymer. And this polymer has lots of pores with carbon dots. Now why it is important to have many pores? What? We need to breathe. We need air to come in. So if we put something without pores, we're not going to have enough air. So it's very important, if we want to have this on our face mask, that it will be porous, that it will let air coming in and out. So we have this thin film of polymer with the carbon dots. And now we put this on a simple cotton face mask, same cotton that we use, and then we can walk outside. When the sun will heat, will shine and put light on this polymer layer, what will happen? There are carbon dots there. The carbon dots are going to absorb light. The light that the carbon dots absorb becomes heat. And viruses, just like any creature in the world, they don't like heat. When you heat them, they will die. Okay? So when you're exposed to sun, you will simply kill the viruses through heating the polymer layer. Now you might ask me, okay, if you have this photothermal effect, you will burn your mouth because it's going to be hot. So that's also not very attractive. But the thing is, you put the polymer on top of the mask. So we actually made these face masks. We use them in the lab. So the cotton is sitting here, you don't feel the heat of the polymer. And the polymer layer is very thin. So it's enough just to heat the polymer layer so the viruses will die, but it does not hurt our body. So this is how it looks like. And again, the same story about photothermal effect of the carbon dots, the heat released in upon a shining light. And this is actually what we got. What we see here is the infrared camera. Remember, these are pictures from an infrared camera that is sensitive to temperature. The more red the color, the higher the temperature. And what we see here, this is the pictures from the camera of the polymer face mask with the carbon dots. And this is just the carbon dots this is just the polymer without the carbon dots. Okay? Now without the carbon dots, see, we heat 20 minutes, we expose to the sun for 20 minutes. After initially 27 degrees, we expose to the sun 5 minutes, we get 33 degrees. After 20 minutes, 41 degrees. Now what happened when we have carbon dots there? We start with 28 degrees. After 5 minutes, we already reach 58 degrees. After 20 minutes, we have 66 degrees. Now, 66 degrees, 66 degrees here is very bad for the viruses. When the viruses experience 66 degrees, they die. People don't like 66 degrees. Viruses also don't like 66 degrees. So, what we get is killing of the virus through the temperature. Now, with a face mask, you want to wear it for several hours. It's not only one time. So what you can see here is that we can actually cycle it. Many, many cycles, 30 cycles. We shine light, the temperature goes up. We turn off the light, goes down. Up, down, up, down. We can do hundreds of cycles. The face mask will be good. Thank you. Oh, questions? Oh, sure.
Hello. Thank you, Professor Raj. I now request the participants to put up their questions, if any. You can ask question if you have.
Yes.
Thank you for your lecture. I want to ask the question that what is the reason for what is the driving force for the nucleation of the metal ions, and why they are converting into so small size? This is a nanoparticle.
Why the metal ions converting to? Again, please ask the question again.
We are using the reducing agent. The metal is going to the zero oxidation state.
Exactly.
Then what is the reason that it is becoming so small to nano size?
Okay, that's a very nice question. I'll repeat the question again, and I hope I understand it. To make nanoparticles, we take metal ions and we reduce them with a reducing agent, and we get the metal zero. Now, the question is, if I understand correctly, you're asking why the metal zero go together and make small nanoparticles? Okay. You know, in nature, it's very rare to find individual metal atoms just floating around. It is not a stable state. It's not a coincidence that in nature you mostly have the ions, because metals like to release electrons. In order to have stability of individual metal atoms, they need to stick together. Then you have the forces between the metal atoms in order to form small clusters, small aggregates, which are the nanoparticles. So thermodynamically, it's more stable to have small aggregates of metal atoms.
Next.
First of all, thank you, sir, for the lecture. Sir, from yesterday onwards, we are talking about the photothermal effect of carbon dots. So, sir, my question is, how sensitive are carbon dots to photothermal effect? Means, what should be the intensity of the sunlight to initiate this entire process?
Okay. You know, carbon dots will always have the photothermal effect. How sensitive it is depends on the carbon dots themselves. Some carbon dots absorb light and release heat more efficiently. In some other cases, it's less efficient. The most important factor for this photothermal effect is how good the carbon dots absorb light. And this depends on what we call the absorbance spectrum. You know UV vis absorbance spectroscopy? There is I wish I could have it, but there is an absorbance of particles of light in different wavelengths. If you take carbon dots that have very good absorbance, very efficient absorbance of light in the visible region, then you have a very strong photothermal effect. Sometimes you have carbon dots that do not absorb so well, you have less efficient photothermal effect. This is the most important parameter. Of course, other parameters is the intensity of light that we use, the concentration of the carbon dots that we use. These are also important parameters.
Sir, in case of the bacteria. Namaste. Thank you, sir, for a nice informative lecture. In case of bacteria, the silver nanoparticle is penetrating the wall of the bacteria and destroying the DNA molecule.
For example, yes.
Yeah, you have shown there. So, is it any evidence like the membrane pore size is larger than silver nanoparticle and they are able to penetrate and go inside and interact with DNA, or it is proposed mechanism?
Actually, we know, scientists found carbon dots inside bacteria. So this is a fact that they go inside. Now, if they go inside, they can do many bad things. Bacteria can take various things inside from outside. This is something well known. There are proteins on the membrane of the bacteria that they take things from outside and they bring inside. Silver nanoparticles are very bad for the bacteria, but they can still go inside and do damage on the DNA and other things.
Sir, very nice lecture. Thank you very much for delivering the lecture. Just like for antibacterial, you are showing the carbon dot with guanidine group.
Yes.
Right? Guanidine group. So that was derivatized, that was synthesized laboratory.
This is a very good question. The answer is no. Actually, the nice thing about the carbon dots is that you can take different building blocks, different precursors, and because you use a relatively low temperature, the carbon dots that you get will contain on their surface the structure of the building blocks. This is really what makes carbon dots so interesting. Because if you take carbon, or these molecules, and you apply very high temperature, you're doing pyrolysis, then you kill all the chemistry. You kill all the chemical structures. But with carbon dots, because you do this in very mild conditions, you get carbon dots with the surface without doing anything else. You don't need to do complicated chemistry.
What's the use of ascorbic citric acid?
Citric acid actually is a well-known component of carbon dots. Most carbon dots, or not most, many carbon dots use citric acid. It's a very common sugar, very cheap, and it gives you the core of the carbon dots.
And the same carbon dots being used for antiviral properties?
No. For antiviral, we use different carbon dots. What we wanted from these carbon dots is that the question that was asked before, we wanted them to have very good absorbance of light. So that's why we used them. It was even simpler carbon dots in the antiviral face mask project.
Thank you.
Or basic question.
Thank you, sir, for the nice talk. So I have a query. Like here, you are using carbon dot as a template for stabilizing silver nanoparticles.
No.
It's on the surface. You are using carbon dots.
What project are you talking about?
Suppose this one.
There's no silver. Just carbon.
And the silver nanoparticles. What are the templates and how is the stability?
The silver nanoparticles is not my work. People just use silver nanoparticles as antibacterial agents without any chemical. Take silver nanoparticles, you add to bacteria, and they kill bacteria through this redox effect, through the electrons on the silver nanoparticles. So there's no chemical.
But silver nanoparticles has some stability issues because it is like degrades in presence of light. So in this case, so in carbon dot case, so what is the stability in presence of light, heat, or other conditions?
They are very stable actually.
So it does not degrade.
I'm sorry?
It does not degrade in presence of light.
Well, after few days or months, they can degrade, but they are very stable. That's actually one of the nice things about carbon dots. They are very stable.
Thank you, sir.
Can we increase the volume? I feel that I'm losing my voice.
One more, the mask you have
Yes.
produced with PVDF,
Yes.
simple,
Okay.
right? And then you are going to embed this carbon dot inside
Yes.
the film. So what's the pore size of this polymeric film?
The pore sizes. The pore size is actually in the tens of the micron range.
So is there any chance when we are inhaling some carbon dot may get?
Excellent question. What we find was that the carbon dots are attached to the surface here very strongly.
Chemically? Or
Actually, no, can be physically, but it's very strong and they don't leach out. They are very stable, so once we prepare them, they stay there. They don't come out.
Good work.
Thank you. I think now the House will undergo 10 minutes recess.
Sure.
And the lecture will resume. The second lecture, today's second lecture will resume after 10 minutes. Thank you.
You know, maybe I'll use this.